1D feature to predict, which is natively unstructured or disordered regions. Let's go a step back. Structure or sequence determines structure, determines function. Right? That's the idea. So in this idea, the central dogma really is you have to fold to a particular, so DNA encodes for a protein sequence for the amino acid, and the amino acid sequence determines how the structure looks, and the structure determines how it functions. And ultimately that's one path. And for this path to work, you need in fact to always adopt the same three-dimensional structure. Now imagine a case such as this one, where we have two molecules, they bind, and the same three-dimensional structure has to work, because if the binding site is not the same, these two proteins cannot interact. Very simple, very clear. Now, it is also true, I've told you, that proteins are dynamic. So here's a particular protein called modulin, and you can possibly already sort of guess that the regions up there and down there will move, will wobble. And this movement, again, is part of the way they act, they function. So this dynamic is built into the structure, okay, the movement. Now there are different movements or different time scales. So ligand binding actually and catalysis, folding goes on the 10 microseconds or milliseconds, hundreds of tens of microseconds to milliseconds, to in fact almost minutes. Allosteric regulation is the kind of large movements. Here is the kind of side chain rotation and vibration. So these are things, the sampling of a different conformation that happens on this scale here. This in fact influences function, but it's not directly visible. It is visible because it produces a sampling of an energy space, and that allows the molecule to bind something else. Anyway, all of that assumes, again, that we have a same three-dimensional structure, and built into the three-dimensional structure is the dynamics, is where it is flexible. Now imagine a situation such as this one here. We have a region here on the left that in isolation is not adopting a regular three-dimensional structure. So definition of not a regular three-dimensional structure, assume I could shine light at it and I could see it. Again, I cannot do that. I need to freeze them, put it into a crystal, and then do X-ray. That's one way, or NMR, remember? But assume I could do that. And then shining light at two different time points, I would see two different things, okay? And that is what we typically refer to as natively unstructured, disordered regions. Upon binding, it adopts a unique three-dimensional structure. Not only a unique three-dimensional structure, but now you're all familiar with this type of secondary structure, I assume. So it adopts a helix. Upon binding, suddenly it becomes a helix. Now, going back to shine light at two different time points, you would see exactly the same. So upon binding, two time points will give the same image. Before binding, two time points will give a different image, because the molecule is too flexible. Here's another way in which you can imagine that this kind of flexibility may help you. What I show is an unbound molecule, and in the bound state you essentially have a long tail. It could be a tail. In this particular case, it's really the end of the protein, the C-terminal end, so the last residues, and the last residues are sort of bound into this molecule. And this in fact is called fly casting. So some of you may be familiar with fishing. So sometimes you throw out a line very fast, have a fly on it, and by that you attract a fish. In this particular case, you attract another protein that you bind to. So in this particular case, the function of this disordered region would be to reach very far. Okay? Like throwing a line. Such a disorder may be, in fact, a good binder. If it is very long, it could sort of wrap itself around something and become a very efficient binder through a large interface. It could be regulated through post-translation modification, meaning that there may be a sugar put onto it, and without the sugar it's sort of bound to itself. With a sugar cut, it is opening up. So by cutting the sugar, you would essentially create a mechanism by which you open this fly-catching mechanism. And in some sense, it could increase the complexity. How could you imagine that it increases the complexity? What else could it do? Well, one idea of increasing the complexity, if you have something, go back to this image that I showed you a few slides ago. In the regular way, you have always a key and a hole, right? The same key fits to the same hole. So a one-to-one match. Now in this example of disordered, you could imagine that the key that you have here, calling the disordered region the key now can fit into different holes. Because it can adopt. It's like an induced fit in an extreme case. And that means increased complexity. It sort of can, with the same thing, bind to different substrates. The regulation becomes more complex. Then you have to make sure that the right things is not at the wrong point at that moment. Okay, so in this sense, we have sort of a coupled binding and folding mechanism. We have order as a scaffold for disorder, and then actually turns out we have a bunch of different types of natively unstructured or disordered regions. So the ones I talked about are just some examples. Another example of what you could do is you could, like in an Amazon package, try to keep stuff away from some other stuff. So you have a glass ball or a glass sphere somewhere inside, and you want to avoid that it gets crashed. So you put some buffer material around it. And this is the same thing here. So a disordered region, you can imagine, like a buffered region, because it samples, it keeps moving a lot, and not adopting to it. So there is the disorder that doesn't bind, and is this type of buffer material. So by moving a lot, it just keeps things at bay. Okay? Another mechanism is a little bit like the fly casting idea, where you can imagine that if you have a long arm, you can sort of sense what is coming around in the vicinity. And sense could be, so in this case of buffering, you keep it away, but sense could also simply be give a signal that something approaches, right? And that again is a mechanism that we observe in disorder. So you essentially use the idea of disorder, I mean, not you, proteins or life, the cellular machinery uses this idea, this concept to detect something that comes close. Now, what's the kind of data we have? There is one database, and I'll get back to that, called DisProt, that collects proteins that have these regions. One particular type of disorder is loopy disorder, and loopy disorder is defined. So I talked about regular secondary structure. Most proteins have about 30% helix, 20% strand, and 50% loops, roughly. But assume you have something where over 70 residues, which is almost about the size of a typical domain, you have less than 5% of the residues in helix or strand. So essentially this whole thing is loop. A loop of 70 residues. The first question that we asked when we came across these things was, how do they look when we look at PDB? And in fact, there was not a single structure known for any of these. We don't have 70 residue loops for which we know the structure. The things that got closest to it, the 4 examples or 4 types shown here, are cases where you essentially have a loop that connects. So we have a very long loop that connects 2 parts. Again, they're not 70 residues long, but the longest things that we saw did something like that. There is the situation where you have very noise, and they will be ignored, at least by the machine learning device that I showed is a neural network, yes?
Is it a different way of saying that error is random? Error is, sorry, there is no pattern.
That is the hope. That's a very good point. So he says, well, I did not quite say that right. I also have to assume that the error has some randomness, that the error is white noise, that there's no systematic error in there. That is partially true. But again, since my machine learning has to put 2 data sets apart, if there were a systematic error, it would just ignore that. Hopefully. As long as the dominant, the consistent right signal is consistent enough, it will be able to simply say, okay, those I cannot classify, and will flop or flip between the two states of sometimes saying this, sometimes not. But if you have enough good data in there, then it will that will determine the extreme answer. So it can even handle consistent, or not consistent, it can even handle systematic errors. And this method, and the next slide I'm going to go over because it's too complex, but what this method can actually do, it can distinguish between a 30 residue loop that looks structured and a 30 residue loop that looks not structured. So it can really, on the same length, it finds things that are 30 residues and that are disordered, and things that are 30 residues and have no regular secondary structure and are not disordered. It really achieves this thing. The great advantage here is I have a large data set, because I can actually predict for millions of sequences, and that's what we do, what we used underneath. And that again was something. Somebody has too many batteries. I want your battery with the power. So the advantage here is that our data was not sharp enough, but we had so much that the system could zoom into the important signal. The third idea is one that has to do with contacts, with contact maps. So disorder is something that is floppy, okay? Now, whenever you fix a certain region with a lot of binding contacts to something else in the protein, it cannot flop. Being flexible means that the part that is flexible cannot be completely constrained. Trivia, right? So, in fact, if that is the case, why can we not just predict contacts? So we look at every pair of residues and predict whether they're in contact. Now this part of the lecture I will not get to in this semester, which is the prediction of contacts. You can do it. It's not good enough to predict 3D structures. I told you that from 2D prediction, or from 2D contacts, you can in principle predict a 3D structure, but these predictions are not good enough for doing that. There are some examples where people have been able to do this now for some proteins. Some is defined as in the hundreds. For those we can predict 3D structure. We have some short proteins for which we can predict 3D structure by molecular dynamics. But that ultimately is the state of the art. But these context predictions are good enough to distinguish between regions that are very constrained and those that are not. And that is what is used in this method here. And in this particular method, you could see that this myosin here is used as a glue and in fact is disordered. There are different types of, different flavors of disorder. And we now have different methods that sort of predict different flavors. The important part is that I have sort of developed a bunch of prediction methods that all go in the direction of disorder, and none of them has really used disordered proteins, has looked up a database of disorder to in fact optimize machine learning. And that's great. If you can get away from with using data in the way we have done it here, you're very safe because there's no cross-validation anymore. You have essentially established a method on some other story. If it works on the next data set that you get, then you have won. So the question then becomes, how do you actually experimentally handle disorder? So first of all, there are different types of disorder, and there are in fact also many different experimental ways of handling it. The first one is what I would call the Dunker hypothesis, which is essentially you look at three-dimensional structures, and in three-dimensional structures there are some residues that you don't see. And the argument from Keith Dunker, and I should have had this photo here, is on the next slide. The argument simply was that the ones that I don't see are disordered. You don't see them because they move too much. So there is no, again, when you do a crystal, you have many copies of the same protein, you shine X-rays at it, and it scatters, and the consistent scattering is what you need. So all the repetitions of the same protein in a crystal have to have the same shape there. And places you don't see is where, in fact, you get an inconsistent signal. Inconsistent signal means movement. And in this particular interpretation, it means disorder. Here is, again, a plot of residue-specific signals for this disorder. in different types of experiments. Clearly there is a difference in in the residues for the disorder, and they initially built a database, I briefly mentioned that already, DISPOT, that uses that. DISPOT also uses data from other experiments, more low resolution experiments, more NMR experiments, where it is measured how regions move. It's very detailed, very complicated, but the body of data is increasing, and that all leads to this database here that annotates disordered proteins. Now, once you have a database like that, you can build all kinds of machine learning devices. And at this point, there are many hundreds of methods out there. Since there's a data set, people apply all kinds of machine learning tools to it. The problem is, again, there's a little bit of overtraining in here, and the field is sort of, in that sense, going in a circle. Now, with the other three methods that I talked about, one way of using this is to just put a meta predictor on top of it, so that combines many methods. I said there are many methods out there. This is just a small set. And then for all of these, you get ROC curves, as you see, that are very close to each other. So this is on the x-axis false positive rate versus true positive rate, and you see that essentially the field is very close to each other. And this one particular method that I talked about in the end here uses the Norse predictions, the contact predictions, the B-value predictions, and some other aspects to slightly improve the blue is slightly higher than what you had before. The main findings is there are different types of disorder. Here are two extreme different types of disorder that have two different functional reasons. I want to skip over that one and go to this finding. So when you look at what fraction of the proteins have at least one long region of disorder, blue and red are two different methods to predict disorder. So you have eukaryotes, bacteria, and archaea. And what clearly hits you is that there's a big difference between eukaryotes and archaea and prokaryotes. Bless you. There's sort of 36 to 43% of all the eukaryotic protein have at least one long disordered region, while the same number for bacteria and archaea is in the ballpark of 7 to 13%. Why could that be the case?
Eukaryotes are more complex, that's why they need more of that disordered region.
So if disorder helps in complexity, is a stepping stone in complexity, then maybe that in fact is exactly what we see here. We see the big jump on the molecular level of complexity, and many people argue that. In fact, we have even written a paper in which we put that into the title with a question mark though. But is disorder one stepping stone in complexity? Possibly that's one reason. If that were a reason, then the following idea that comes as molecular recognition element, again comes from Keith Dunker originally, or the more, where you have a particular element that sort of it can change its structure, and by adopting a certain region it binds, it doesn't bind, it becomes a glue. And this glue could even be bring two proteins together in a sort of more complicated brace kind of thing. It could be like a brace that connects to different molecules. And this is exactly what has been observed. If you do this, you add an interaction between two proteins that otherwise you wouldn't be bringing together, meaning you allow proteins to have more interacting partners, and that intrinsically increases complexity. And that is so there's a lot of evidence for that. Another way of looking at it, when we look at the bacteria and we ask, well, is there a difference in the disorder content between bacteria? So bacteria have much less disorder. Do they differ? And the answer shown here is yes, they differ. So in the top, below is by evolution, so by things are close to one another that are close in terms of evolutionary relation. Up there, close in habitat, meaning they are extremophiles, so there are bacteria that live either in the heat that can sustain cold, that can sustain very salty environment, or very hot environments. And it turns out that in fact, bacteria that live in extreme situations are most similar in their disorder to other bacteria that live in the same environment, much more similar to those than to their evolutionary closest homologs. So the bacterium that is closer in evolution is different in disorder than the other bacterium that is very far away but has a similar environment. So disorder has to do with environment, is what that one shows. And that is remarkable because it's a very, very simple feature. You just look at the overall content of proteins that have one disorder region over 30 residues. It's very, very simple. Okay, now here's another rather peculiar experiment typical for biologists. So they take yeast and they torture it. So they put it into all kinds of pressure situations. They take food away, and one particular pressure environment is heat. So yeast likes a certain environment which is close to the room temperature, and they just put it into 80 degrees or into very dry environments. And then they see, does any of the yeast survive? And most of them die, right? And some of them survive. And these that survive, they select. And then, they do that again and again. They actually do that 1,000 generations. Just, 1,000 generations of people is sort of a long time. Do the math, 30 years per generation, 30,000 years. This is close to the existence of maybe we are exist 10 times longer. Maybe Homo sapiens is 100,000 or 300,000 years old, as the last week report says from Leipzig. Maybe we are 300,000 years old, but that's still only 10 times what they do in this experiment. Actually, they go further. So what do you believe? So they have survivors in the heat. What do you believe is the reaction of the ones that survive? How do they survive? Of course, then they go and see what's the molecular mechanism. So what is different between the ones on the molecular level? What's different between the strains, or between the ones that survived and the ones that didn't? What kind of mechanism has happened or changed?
Some kind of mutation?
Some kind of mutation, yes, that's true, but what kind of mutation? So, in fact, the funny thing, there's a fast answer and there's a long answer. So the fast answer is for 1,000 generations. After 1,500 generations, there is a longer track solution. The solution after 1,500 generations, so if yeast had time enough, they really change the proteins that they express. So they in detail found the proteins that they need to express more in order to sustain the heat, okay? But for the first 1,000 generations, there's a rapid reaction that is not at all as specific. And that simply is the duplication of an entire chromosome. Now, for me, this is remarkable. So, yeast has 16 chromosomes, and so you duplicate 1/16 of your entire genomic mass in every single cell that you produce to survive, and very specific. So we asked, what is so specific about this chromosome that is repeated? It's always the same chromosome. In fact, it's chromosome 3, shown in red here, and it turns out that chromosome 3 has very low disorder, and in fact, when you look at when you look at the chromosome that is not too big, then this one is the one with the lowest disorder. So you want to duplicate something that is not too big, not too small, and that's the only one that you get. So that's not causal. So disorder has something to do with why the heat resistance is created, but we don't see a causal reason. In order to see a causal reason, what you have to do is map all the proteins on that region that is duplicated to what they do. Now we run into the problem that, unfortunately, not for all proteins we know what they do. So we cannot take an entire chromosome, or we can take an entire chromosome, and for 40% of that chromosome, of the proteins in that chromosome, we know what they do. So there's 60% unknown, and we hope that the 40% that we are now going to look at contain all the truth. Okay, now what we do, we take this 40% for which we know the function, and we ask, how is that function different from all the other things? What is enriched in that set? And the answer of that one is everything that has to do, I'm sorry for the size, it's not readable, but the answer is everything that has to do with sexual reproduction. And that makes sense, right? You're under stress. It's better if that group of yeast can reproduce more easily under a stress scenario, that clearly is an evolutionary advantage. Makes a lot of sense. And we felt, bingo, we nailed it. So we have the causal reason here is really that on that chromosome you have things that help the carrier to survive. Unfortunately, this particular reproduction of yeast was not sexual. So yeast has two ways of reproducing. One is asexual, and that's what these guys did. So we have no idea. So from all the proteins for which we know function, that's the only significant difference. And that significant difference does make sense biologically. So that's a dead end. And this is the typical kind of story that we often go into or run into or fall into in biology. So from what we see, what we observe, we cannot explain what we see. The only thing that remains consistent is this story about disorder, but their causality is the problem. And with that, we get into the issue of causality.